Hello, immunology fans. With the last discussion that we had on complement, there were just a couple slides left on that, and I will come back to them later. This flipped lecture will be on the beginnings of what is acquired immunity and what are the receptors and how are they generated and how they generate such diversity and specificity. The big difference between innate and acquired immunity are specificity as compared to relatively nonspecific PAMPs, or PAMPs that can be found on multiple different pathogens that are recognized by receptors that are on multiple different cells, and memory. Memory meaning that once you see and make a response to something, you now have generated cells that will remember that and will be able to protect you in the future. The two different kinds of receptors that we're going to talk about are B-cell receptors. And B-cell receptors are found on the surface of B cells, and they literally are antibodies. And so they are called surface immunoglobulins, or surface Igs. B cells, any individual B cell will express surface immunoglobulins that are specific for only one antigen. So on this cell shown on the right of the slide, the two B cell receptors, or surface immunoglobulins, they're antigen recognizing domains that I've just circled. They all recognize the same antigen. So one B cell can only express one antigen-specific surface immunoglobulin. These B cells, when they differentiate, and we'll talk about that later, they become terminally differentiated into something called a plasma cell. And plasma cells will now secrete those immunoglobulins, and they are then called either antibodies or immunoglobulins, and they are no longer surface immunoglobulins or B-cell receptors. Differentiation, in this case in B cells, results in the same antibody molecule now being made and exported out of the cell as a soluble protein to help in your specific acquired immunity. The other type of cell receptor that we're going to talk about are T cells, and T cells have T cell receptors, TCRs, so BCRs and TCRs, and each T cell expresses its T cell receptor with only a specific single antigenic activity. So just as B cell receptors can only, on any individual B cell, will recognize one antigen, T cells, every T cell, will recognize one antigen. First, we're going to focus on antibodies, or surface immunoglobulins, and how they get made and what their structure is. On the bottom right in this image, you see the structure of an antibody. Each box in the antibody, so you should see two heavy chains, and I'm circling the heavy chains. So one heavy chain on the left and one heavy chain on the right. Each heavy chain has 3 blue boxes and 1 red box. So the heavy chains have 3 plus 1, and each light chain, now being circled. All right. So the light chains have 1 plus 1. And so every time you see this box, you are going to know that it is a similar fold in a protein. It's a domain. And this particular domain is called an immunoglobulin-like domain. On the left-hand side, is the ribbon diagram for the picture shown on the right-hand side, which is just a schematic diagram. And what you can see, I hope, is if you follow the yellow colored ribbon, you can see that it has some beta pleated sheets, which I just drew a black arrow to, which may not be all that visible, that are aligned in a particular way. And every one of these domains in the yellow all have that same beta pleated sheet structure. Similarly, the blue heavy chain has these 4 similarly folded immunoglobulin-like domains. Each light chain has 2 of them. On the right-hand side, where I've shown you the heavy and it says 3 + 1, the 1, even though for each of the heavy and light chains, even though they have the immunoglobulin-like fold, they also have the capacity to bind antigens. Okay? The purpose of these proteins, right, this is just proteins, and this is a quaternary structure that's put together when you have more than one polypeptide chain, is to, when it's a surface B cell receptor, it can recruit other cells and molecules to destroy the pathogen, and it can also result in signaling. And we're going to talk more about that momentarily. An additional way that these red boxes are known, and because they bind to antigen, is they are called the variable regions of these molecules. So each heavy chain and each light chain has a variable region. Cleverly, the other regions are called constant regions, as I'm circling on the bottom right. So the heavy chains have 3 constant domains, and actually some antibodies have more. And each light chain only ever has 1 constant region and 1 variable region. A little more specificity here shows you that each antibody structure has 2 heavy chains which are 50 kilodaltons in molecular weight. Those heavy chains are linked together in what we call the hinge region. So the hinge region, which I've just circled here, they are bound to one another by disulfide bridges. So this is where we have disulfides. We can actually get covalent linkages between the 2 heavy chains. Similarly, there are disulfide bridges between the upper regions of the heavy chains and the constant regions of the light chains, circled here in red. The light chains are each 25 kilodaltons. So the overall molecular weight of an antibody is 2 times 50, which is 100, plus 2 times 25, which is 50, which equals 150 kilodaltons. The disulfide bonds are made where we have what amino acid? Anybody remember an amino acid that allows for disulfide bridges? It's cysteine. All right, so when you have a cysteine, they have the sulfur at the end, and they can create the disulfide bonds. And as I said, in this case, the disulfide is in the flexible hinge region, and literally it's flexible. The protein can actually move somewhat. And so there's 2 disulfides there, and then there's also disulfides between the heavy and the light chains. Well, there's more to it than that, because every time you make a heavy chain, it can be paired with any light chain. All right, so let's say you make heavy chain A, and then you make some light chains. The light chains can have different variable binding regions, as well as the heavy chains. And so let's say you have light chain A, light chain B, light chain C. You get diversity by linking any individual heavy chain with any individual light chain. On top of that, there are two varieties of light chain. We call them lambda and kappa. So you might have lambda and kappa, light chain A, B, and C. So not only are you getting combinational diversity by combining different light chains with heavy chains, you can also get combinational diversity by simply linking a heavy chain with any individual lambda or kappa. All right? On any individual antibody, you never mix lambda with kappa, meaning that as I draw this antibody on the bottom, I'm making two heavy chains. And for the purpose of this, the light chains I'm showing you are in blue. And if you have lambda, they're both lambda. If you have kappa, all right, so here I'm drawing another one. If you have kappa for both. You cannot mix the two. You cannot have a kappa and a lambda light chain. This does not happen. Let's draw a big circle through that and say, no, does not happen. Interestingly to me, different species express different ratios of kappa to lambda. Humans are relatively close, a 2-to-1 ratio with more kappa than lambda. Cattle have a 1-to-20, which means more kappa to lambda, and mice are exactly the opposite. Why will become apparent perhaps momentarily. Well, you might have thought that was the end of the structure. No, we're just going to drill down, and we're going to drill down, and we're going to drill down, because it really is the structure that leads to the function in this antibody structure. Okay, so in this image, what you can see now is they've labeled the different constant regions. So each heavy chain in this image has constant heavy 1, constant heavy 2, and constant heavy 3. I won't ask you whether 1 is at the bottom or 3 is at the bottom. I don't really care. But each of those is one of those immunoglobulin-like domains. Similarly, we call, right, so there's going to be a variable region for the heavy chain. Then for the light chain, you just have one immunoglobulin-like constant region for the light chain and one variable region for the light chain. And remember, the light chain will be either kappa or lambda. There are 5 different classes of antibodies, and they each have somewhat overlapping but distinct functions. And the classes are called IgM, IgG, IgA, IgD, IgE. Each class can have additional subclasses, and it's species dependent. So in humans, there's IgG1, IgG2, IgG3, and IgG4. It gets complicated, all right? And those different classes are called isotypes. So if you see IgG1 versus IgG2, they're different isotypes. They still have this structure. And do you remember when I said some immunoglobulins have more than 3 of these constant regions? The 2 that have one more constant region are IgM and IgE. I always remember this as me. They actually have constant heavy 1, constant heavy 2, constant heavy 3, and constant heavy 4. One last thing to note on this page, the different heavy chains are called by their Greek, is that Greek? Latin, I don't know, letters, mu, lambda, alpha, delta, and epsilon. So you'll see Ig mu or something like that, and they're talking about the IgM heavy chain. Structure is so much fun. I really love it, so I wanted to show you this picture, which actually gives you more information about these immunoglobulin-like domains within any individual heavy chain. Each immunoglobulin-like domain, circled here in blue I'm showing you, they all have disulfide bonds within them. So the light chains have disulfide bonds. There's interchain and intrachain. Inter is between chains, and intra is within any given polypeptide. Alright, so there's a lot of disulfide bonds that allow for the structure and the function of these proteins. If you were to put these proteins in a reagent that reduces disulfide bonds, the entire thing comes apart. Alright, generally light chains have 2 domains while the heavy chains have 4, and they actually have 5 if you're talking about IgM and IgE. And I'm wondering if you want to think about what that means with respect to the evolution of these proteins. When you see a lot of the same thing happening in an evolutionary perspective, what it suggests is that something worked and that it was structurally advantageous. And for whatever reason, these structural folds were duplicated in the genome. So you get duplication of parts of the genome that allow for additional domains to be found in any individual proteins. Often you might get a string of the same domain, as we had seen. Perhaps you noticed it in the NOD proteins. There are also these types of things in proteins that link cells to cells, such as ICAMs. And so evolutionarily, think about this as a molecule that quite likely was successful early on and tended to increase the number of domains because of this genetic duplication. All right, if you look at the ends of each of this antibody molecule on the right, what they're showing you in the black, all right, which I have just circled in blue, is what we call the FC region, and that stands for crystallizable fragment. And what they're talking about there is they were able to take that portion of the protein, they actually took off the remainder of the protein. If they express that part of the protein, they are then able to crystallize it and look at the structure. When they crystallize proteins, it's a particular process that literally allows crystals to form. And you cannot crystallize proteins that have any kind of sort of mobile elements in them. And so the whole antibody molecule can't be crystallized because it has this mobile region, the hinge region, which allows for flexibility in the protein itself. All right, so the bottom part is called the crystallizable fragment, or the FC region. And at the very top, then, they're showing you what they call CDRs. CDRs are complementarity determining regions. And these complementarity complementarity determining regions. I don't really like where they've put the arrow. I would suggest to you are the ends, right, of the proteins, and I'll show you in the next image why that is the case. Because even though they're showing you that the N terminus is at the very end, that's not very true. In the folds of the immunoglobulin-like domain, you're going to see that there's loops in this structure, and it's actually loops within that immunoglobulin-like fold which create complementarity, and they're complementary to what they bind to. So if you think of it as maybe they create little barbs, that's going to allow it to bind to something that has, grooves for those little barbs. It's not a very good picture, but think of it as a key and a lock. The key fits in the lock, and these complementarity determining regions are the key that can go fit in the lock of the antigen. All right. Interesting research was done to examine the different domains, and specifically because they were trying to do this crystallizing to see what the structure looked like, they used different enzymes to cleave the protein, so different proteases. So they looked at proteolytic cleavage, and they used, in this case, an enzyme called papain, and that enzyme happened to cut just above, so here, the papain cut just above the disulfide bond in the FC region. And consequently, you are left with these FC regions which have a very nice, strong structure to them. And that was the portion that was crystallizable. The other portions, which are actually separated now from one another, are called the FAB regions. And they called them this, right? So I think it's based on the fragment, and it's the antibody binding fragment, and each antibody has the ability to bind an antigen. And so the FAB fragment is the fragment here on the right hand side that I just drew an arrow to, so there's two of them, so that when you cleave using papain, you end up with two FAB fragments. Each one can bind to antigen. They both bind to the same antigen when you cut any individual antibody. All right. They used another enzyme, and the enzyme they used was pepsin, another protease, and this protease cleaves multiple times in either the hinge region or in the FC fragment. And what that does is it results in basically degradation of this FC region. I'm not sure that anybody uses this little piece down here for anything, but we have been able to use this top piece for particular experiments. And this top piece, now, the big difference between cutting with papain and cutting with pepsin is that when you cut with pepsin, you end up keeping the disulfide in the hinge region, along with the disulfides in the light and heavy chains. And this creates basically the two arms of the antibody. And this will allow the FAB, and they call it FAB prime 2 because it has two antigen binding sites, they stay together and they can be used in experiments. The reason it's important that we discovered this, well, we figured out, I'm not sure whether it was using these or by another technique, but we figured out that you cannot activate B cells by binding an antigen to an individual receptor. You actually have to bind an antigen, and this is shown on the left here, which I'm going to blow up. You can only activate signaling inside of a B cell when you have antigen that binds and cross-links the receptors together. And this is because you're going to see there's a lot of stuff happening between the molecules bound to the tails of these receptors. And when they get brought close enough together, they can signal. If you have an antigen, and so if I put my own antibody out here, all right, here's an antibody, and I put, I don't know, an ugly little antigen here. If it only binds to one antibody and it does not cross-link for whatever reason, it will not cause signaling. And I believe that they used, by comparing what happens when you add FAB fragments versus the FAB prime 2 fragments, right? FAB fragments would cause no signaling, whereas the FAB prime 2, yes, it causes signaling. Okay? So understanding the structures is really important because it gave us an idea of how the function is downstream of antigen binding. You know, antibodies are so important in so many different places in your immune system. The idea to genetically engineer antibodies for therapy has been going on for maybe 10 to 15 years, and this is probably old school now, but they were trying to make something called a single chain FV, which is basically, you know, the variable fragments. And instead of having to make two different proteins, instead of having to make the heavy and the light chain, what they tried to do was they took what they knew from a heavy chain. So they knew that you could have, right, a domain here that had antigen binding capacity, and it folded into a immunoglobulin-like structure. And similarly, the same thing with a light chain, immunoglobulin-like variable region. And literally, they took it and in the very bottom right here, I hope what you can see, oops, I have that arrow going to the wrong place. I'm going to erase that. All right, so here is the variable region from the light chain. Here is the variable region from the heavy chain. And they took those two DNA pieces and linked them together with additional, we call it linker sequence, and tried to generate these, I think, SC soluble FV therapeutic antibodies. And so this is not perfected yet, but it definitely is well down the pathway for being able to be used. There will be additional ways of genetically engineering lots of different molecules in the future, and your generation is really the ones that are going to make advances in this. We know a lot now, but you're going to know a lot more. Oh, structure is my joy and passion, and this really gets at how you can have the same exact folds in all of these regions. These are all immunoglobulin-like folds, but you can also create diversity and the antigen binding sites, or the complementarity determining regions. And it has to do with where those amino acids are in the sequence. So let's start looking at these images. Let's look at the bottom right image. Let's look at the light chain variable region. All right, I'm circling what I am looking at. And literally what they're showing you here on the very bottom is just a line, right, a box, and they're showing you where different regions of the final protein lie. And in the box, so they have FR1, HV1, FR2, HV2, FR3, blah, blah, blah. And I actually don't know what FR means, but HV is hypervariable. And so you see that there are hypervariable regions in 3 different places. And if you look at the image directly to the left of that, in both the heavy and light chain, you have 3 hypervariable regions in the amino acid sequence. What that means is, all right, we're talking about methionine, cysteine, glutamic acid, lysine, serine, blah, 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 blah. Well, in both the heavy chain and the light chain, between amino acids 0 and, let's just say, 0 and 30, which I've just underlined, and they're not exactly the same spots, but they're pretty darn close. Between 0 and 28, 29, 30, there's not tons of variability in the number of amino acids that could be there. And so variability, how many different amino acids can you have? Well, you could have 20 different amino acids. So this is a little misleading because they're saying variability is 100%, right, or higher. Well, the variability isn't based on 1 to 20. They're saying if 20 different amino acids could be in that position, I suspect that's the highest variability it could be. Don't worry about the numbers, how many are different. Worry about the fact that in each of these heavy variable chain and light variable chains, you have 3 regions that have lots of variability, because those are the spots that are going to be binding to antigens. All right, so the bottom right is just saying, look, there's places where we have lots of variability, and that's going to create specificity for antigen. On the bottom left, bottom of this, go to the bottom left. Now what they're doing is they're showing you, all right, let's look at the yellow and the green. In the yellow and the green, if you start at the beginning, which I'm drawing a red arrow to, right there, that is the N-terminus of the yellow chain. And remember, I told you it makes beta-pleated sheets. So in this case, it makes a beta-pleated sheet, and then it has a turn. Right, so I've just put an arrow to the first turn. It makes a beta-pleated sheet, and it makes a pretty wide turn. All right. Then it goes over to the right-hand side where it's a beta-pleated sheet, and it makes another wide turn. All right, and then a beta-pleated sheet, turn, beta-pleated sheet, blah, blah, blah. That's what you get when you have these beta-pleated sheets. All right, great. Sounds like fun. Where in God's name are the hypervariable regions? Well, the hypervariable regions, all right, I'm trying to see on the top, are in loops. All right, on the light chain I can see the loops. So I'm drawing a red arrow to 3 different loops. Those 3 different loops correspond to the 3 hypervariable regions in the light chain. Similarly, there are 3 hypervariable loops that correspond to the hypervariable regions in the heavy chain. And it literally is those loops that interact with the antigen. Pretty cool, yes? This picture shows it way better. This is showing you heavy and light chains. And so you are going to have these complementarity determining regions. And you're going to have it for the light chain, and you're going to have complementarity determining regions for the heavy chain. And each one has 3. So you're going to have 1, 2, and 3 for the light chain, and 1, 2, and 3 for the heavy chain. And so you get all 6 of those coming together, shown here, as those loops are what can be different and create specificity for binding to antigens. Now that is cool. Okay. In this case, all right, I said that you could get combinational diversity by mixing heavy plus light. You can also get combinational diversity by mixing heavy plus lambda versus kappa. Still light. Well, now you're going to get tons and tons and tons of variability based on different complementarity determining regions. Okay, lots of diversity, and that leads to incredible specificity in the ability to recognize lots of antigens. Okay. This picture shows it a little better. It shows you the hypervariable regions along with where those hypervariable regions are in the bottom left-hand image. And so they're on the top shown in red, and they exactly correspond to what is shown in the top left image, these hypervariable regions within the amino acid sequence. Pretty darn cool. Well, we haven't talked all that much about antigenic determinants. What are antigens? And anything that can be recognized by an antibody is called an epitope. And epitopes are pretty darn small, and so most large molecules can have multiple epitopes and can be recognized by a whole bunch of different antibodies. And antibodies don't just recognize proteins, they can recognize polysaccharides, they can recognize nucleotides, and we talk about this they either can recognize what we call conformational epitope or a discontinuous epitope, and I'll talk to you about what that is in a minute, versus something that's continuous and linear. Continuous and linear is easy to imagine. I'm talking about a string of amino acids. All right, so let's just say it's serine, threonine, alanine, phenylalanine, leucine, methionine. All right, and it's going to be slightly longer than that, but I don't want to say any more. So a continuous or a linear epitope is literally just a string of amino acids. And that's a drastic difference from something that could be created by loops that are far apart on any individual protein, and that would be generated based on the conformation of the protein. And so an antibody could recognize that particular conformation, and it would only recognize it if the protein was not unfolded. And so those are called conformational epitopes. I won't use discontinuous. I like conformational versus linear. Antibodies can be made to both. Well, how do antigens and antibodies interact? It's all non-covalent, all right? It's nothing more than the normal non-covalent bonds that you know. There's hydrogen bonding, there's hydrophobic force, there's ionic bonding, and van der Waals. A lot of these are based on ionic bonds, and so we know that high salt and differences in pH can change them. To break these bonds, you don't have to do all that much. Detergents will undo them. And to be honest with you, once you start messing around, detergents, high salt, and pH can also denature the proteins. But binding between antigens and antibodies is reversible, and it depends on the strength or the affinity of those interactions, how long they will stay together. Okay, so that was a B cell receptor, lots and lots of information, more than you probably wanted. We're going to talk about the same kind of deal with T cell receptor. Okay, let's talk about the T cell receptor. T cell receptors only ever show up on the surface of a T cell. They never get secreted. T cells generally only recognize small peptide fragments, linear fragments. They're not good at conformational epitopes. And T cell receptors have to be used in combination with what we call major histocompatibility complex. And there's two MHC molecules, MHC1 and MHC2. Excuse me. And the MHC molecules, pretend this is a cell surface and this is an MHC molecule. Alright, here's my MHC molecule. It actually binds peptide specifically, and that peptide is presented to a T cell receptor on a different cell. And the T cell receptor has to recognize both the peptide as well as the MHC. And I'm not drawing that at all correctly. There's additional molecules that do this, but the idea is that T cells recognize peptides in combination with MHC. All right. What's nice is all that structure that you just learned, you're going to be able to, with respect to hypervariable regions and complementarity determining regions, you're going to be able to use it for T cell receptors as well. Here is the image of the antibody comparing it to the T cell receptor. I hope what you can see is that the T cell receptor has these boxes on the bottom right here. Those are immunoglobulin-like domains. All right. Similarly, you have a constant region and you have a variable region for each one of these. And there's two different polypeptides in this T cell receptor. So in this TCR, you have an alpha chain and you have a beta chain. Each alpha chain will have a variable region and a constant region, and the beta chain also has a variable and a constant region. There is a disulfide bond linking the two chains, alpha and beta, and the antigen binding sites are generated by the same hypervariable amino acid regions in these polypeptide chains. Each T cell, right, will have about 30,000 T cell receptors on a surface. So this is T cell receptors on the surface. And any individual T cell will only express a T cell receptor that recognizes one antigen. Just like B cell receptors, they can only express one antigen-specific antibody or surface immunoglobulin. T cell receptors, same thing. We will talk about this momentarily, but in addition to an alpha and beta, there are also gamma and delta T cell receptor chains. And these are used early on in development, and gamma and delta T cell receptor chains are expressed together. And when you switch over from gamma delta, you go to alpha beta chains. So alpha beta chains are what we see after some developmental portion of life where all of your T cell receptors become alpha and beta. Big differences between T cell receptors and B cell receptors. T cell receptors have only one antigen recognition site, whereas antibodies, right, if I just draw a silly antibody, has two. And T cell receptors never get secreted, whereas the surface immunoglobulin, or BCR, can, right, when this cell differentiates into a plasma cell, has the antibody secreted from that cell. More structure. As I said, these are immunoglobulin-like domains, so two yellow, two blue. They have constant regions and they have more variable regions, just like we saw. There is the disulfide bond, and there is a very short transmembrane region. Notice the cytoplasmic tails are incredibly short. I forgot to mention, in the B cell receptor, cytoplasmic tails incredibly short. And that matters because think about if you have something really short, it may not be able to interact with the things that it can excite. And so in this case, we're interested in exciting downstream signaling molecules, and if it's not long enough, it can't actually get the excitation going. All right. So I told you about the continuous versus the discontinuous epitopes, or the conformational versus linear. Remember, conformational is when you have a structure that's created by the folding of the protein. B cell receptors can recognize both. All right, so B cell receptors are super duper. They recognize conformational as well as linear. Generally, T cell receptors, not so good. Mostly short, contiguous peptide sequences, so linear. And often that means that proteins have to be completely unfolded and chopped up into peptides so that they don't create any structure. And this makes sense because I just told you that MHC is going to present peptides. Well, those peptides, all right, so here's an MHC, right? It could present something that's linear, or it could, and that's not very linear, right? But it could present something that also has a little bit of a shape to it. Not much, all right? We're talking virtually linear all the time, all right? So just go with linear for T cell receptors and both for B cell receptors. All right, I'm going to stop there because that's enough for this flip lecture, and we will pick up here with MHC on Monday.